New long COVID trials aim to clear lingering virus and help patients in dire need. In September, Shelley Hayden, a COVID-19 patient, joined a small community of patients at the University of California, San Francisco, UCSF. She had been afflicted with the disease's sequel, long COVID, which causes extreme weariness, difficulty thinking, shortness of breath, gastrointestinal issues, and migraines. There are no proven therapies for long COVID. However, that September day, Hayden documented her optimism for posterity and joined a monoclonal antibody research group. These clinical trials are the first to tackle the syndrome's underlying biology, and several focus on a fundamental topic: is the remaining SARS-CoV-2 virus causing the patient's symptoms? Some people with long COVID do not completely eliminate SARS-CoV-2 from their bodies, as viral reservoirs can be found in the stomach, blood, and other tissues. Scientists want to know if eliminating the virus provides comfort. The trials are few in number and minor in scale, with only 30 individuals participating. Each enrollee carries clues concerning long COVID that doctors hope to uncover. Even if a technique fails, the knowledge obtained may propel science forward. Stephen Deeks, a UCSF infectious disease expert who started researching long COVID in the spring of 2020 and helped design the antibody study, describes long COVID's impact on the body as chaos and mayhem, and is skeptical of what most of these early experiments can achieve. Long COVID lacks objective measurements such as tumor size, brain abnormalities, or blood cell count, making it difficult to establish whether a treatment is effective. Relying just on symptoms can be problematic because they change unexpectedly over time. Doctors are working alongside scientific progress to weave discoveries into trials, which look for therapies and long COVID signals in the blood or elsewhere. The pressure is intense, and researchers are concerned about the pandemic's potential to cause chronic sickness from the beginning. In April 2020, Upinder Singh, an infectious disease expert at Stanford University, was conducting an acute COVID-19 study with a colleague who worked in myalgic encephalomyelitis chronic fatigue syndrome, MECFS, an undefined chronic condition presumed to develop following an infection. Singh pushed the question away, and within a year, Stanford had established a long COVID clinic. According to recent estimates, long COVID affects at least 17 million people in the United States and millions more in other nations. Long COVID is not the first condition to develop after infection, but it provides a unique research opportunity because clinicians usually know when patients were infected and that SARS-CoV-2 was the cause. Scientists began investigating its biology, observing immune system anomalies and blood clotting malfunction in adults and children. Autopsies of 44 persons who died from COVID-19 between April 2020 and March 2021 revealed viral RNA throughout the body, including the lungs, gastrointestinal tract, neurological system, and muscle, months after infection. People with long COVID had similar findings in their blood, GI tracts, and feces. Diane Griffin, a virologist at Johns Hopkins University, has long been interested in RNA viruses, including SARS-CoV-2. Measles virus persistence is often seen as advantageous, acting as a form of permanent immunity. However, it can result in a rare neurological issue a decade later, which is considered to be caused by a dormant virus in the brain. This double-edged effect may be true for many other RNA viruses. Persistence is also seen in long COVID patients across investigations. Harvard University scientist David Walt and his colleagues found SARS-CoV-2 proteins in 24 blood samples from 37 persons infected with long COVID. In February, a large population study in the United Kingdom found that patients with persistent virus in their nose or throat at least 30 days after infection were 50% more likely to develop long COVID symptoms 12 weeks later or beyond. Experts believe that viral persistence may help prolong symptoms, as viruses or viral RNA in the body's tissues may generate an immunological response, such as chronic inflammation or other abnormalities in immune cells. If the virus persists in the gut, it might cause symptoms such as gastrointestinal problems. Peter Brodin, a pediatric immunologist at the Karolinska Institute and Imperial College London, believes that virus persistence is at the heart of all the other problems of long COVID, including a dysfunctional immune system. Some therapeutic trials focus on immunological dysfunction, such as the rheumatoid arthritis medication baricitinib, while others are directly targeting the lingering virus. Paxlovid targets viruses that replicate quickly, which may or may not be the case with the virus found in long COVID instances. Given the uncertainties, some researchers are ready to put more diverse weapons to the test, such as Truvada and Cellzentry, two broad-spectrum antivirals licensed for HIV treatment. Lael Yonker, a pediatric pulmonologist at Massachusetts General Hospital, is currently enrolling 48 children with long COVID and detectable spike protein in their blood for one of the few pediatric long COVID trials. She discovered that medication called larazotide tightened cellular connections in the gut lining and prevented a SARS-CoV-2 protein termed spike from entering the bloodstream, alleviating their symptoms. Larazotide has also been investigated in celiac disease. Another technique is to give monoclonal antibodies that bind to and kill any remaining virus. In October 2023, the American Journal of Emergency Medicine published an intriguing article on this strategy, which described 3 long COVID patients who were given a COVID-19 monoclonal antibody months after their initial infection, either to cure a second bout of the virus or to avoid sickness after being exposed. What occurred next was remarkable, as long COVID symptoms subsided, and all 3 reported good health 2 or more years later. Deeks expects at least an echo of this achievement in the antibody trial performed by his mentee, an eager infectious disease doctor named Michael Peluso. Both physicians were deeply involved in the study of another virus, HIV, and are now conscious of the lessons learned from its history. Deeks believes that finding a similar tracking measure in long COVID is crucial, as it is very challenging to run trials without it. A biomarker would also aid patients by providing validation. There is optimism that the virus itself will serve as a biomarker in trials, but quantifying it in persons with long COVID is challenging. Blood is the best source of biomarkers, as it contains very little SARS-CoV-2. Some groups are looking for viral RNA in blood, while others, such as the NIH's Paxlovid trial, are using Walt's technique to detect trace amounts of viral protein there. Walt recently tested Simoa technology, which was licensed to a firm he co-founded, on thousands of long COVID patients and healthy people. Early findings suggest that approximately 20% of these patients have detectable virus. SARS-CoV-2, a virus that binds to gut cells, has been found in persistent infection in some cases. However, gut cells are fragile and do not survive well in culture, making it difficult for scientists to grow them and investigate their function. Researchers are establishing a mouse model of chronic gut infection with SARS-CoV-2 and researching the virus in human feces samples to better understand which cells it may hide in, how prevalent it is, and how best to detect it. Improved detection combined with immune system analysis has the potential to clear up uncertainty in current test results. Some long COVID patients have detectable virus 1 month but not the next, and many do not have any at all. Current technology may be insufficient to detect it, or the virus or viral RNA may be hidden in inaccessible tissues such as nerves or the brain. It is also possible that some patients with long COVID clear the virus while still suffering from the abnormal immune response it triggered. Following the unknowns, few trials aimed at viral persistence limit entry to those with detectable infection. Instead, they measure it as best they can throughout the study and investigate a therapy's effectiveness in various ways. Researchers are monitoring patients' immune systems, examining cells before and after treatment. Despite the progress of clinical studies, researchers experience frustration and longing for more support from researchers and donors. Drug company investment might provide a significant boost, but many people are hesitant to invest because they are unsure of how quickly they would get a return on their money. Gallons of monoclonals developed by drug companies to treat now extinct COVID-19 variants sit unused, even though they might be tested in long COVID patients who were initially infected with those variations. Studies that fall short should inspire investment rather than dampen it, researchers stress. This year, researchers plan to conduct trials with HIV antivirals, a monoclonal antibody, and medicines that affect the immune system and target clotting problems. By 2026, they hope to establish a trial network to explore therapeutic combinations. For people living with long COVID, the syndrome's complexities, its volatility and grip on so many regions of the body, are unavoidable. Cole Riley, a 9-year-old son, participated in Yonka's experiment in August 2023, after blood tests showed the presence of SARS-CoV-2 spike protein. Riley and Hayden are experiencing similar pain as their child Jackson, who is unable to walk and is unable to do anything about it. Riley believes the experiment is what she was looking for, a ray of hope. Hayden, who has also experienced pain, made a second video after a long infusion day, expressing her hope that the magic solution she received might be kicking COVID's ass. The video ends with the appropriate words, "To be continued." Both Riley and Hayden are unsure if they received the experimental treatment or a placebo.